All right, we're live. I hope everybody can listen to us. Today, Harris will be presenting.
Harrison.
Harrison, I'm sorry, man. I don't know what's happening to me.
It's all
Harrison.
good. Yeah, 3 syllables.
All right, Harrison will be presenting today. I really like this paper. Harrison, if you click on the share screen, you should be able to. Yeah, you should be able to present it.
One Microsoft PowerPoint share.
Sorry, I was muted. Jatin, I think we should introduce our new members first.
Oh yes, and
Yeah,
today
yeah.
we have two new members joining us. One is, so tell me if I pronounce it wrong. Eugenio.
Yes, it's correct.
Okay, and Natalia. Hola a todos los que hablan español.
It's perfect.
So everyone can see my background?
Yeah.
Yeah,
Yes.
looks good.
Cool. Awesome. Look at me. Technologically advanced. So do we want to start with a just real quick introduction of everybody and what they're doing, or do you want to jump straight into the paper?
No, that's actually a quick introduction since we have new members joining us.
Yeah.
I'll start with myself. I am a fourth-year PhD student. My work is mostly around regulatory T cells and a molecule called SOX, suppressor of cytokine signaling 1. It's pretty cool stuff.
SOX1. Yes, sir.
Harrison, can you go next?
Yeah, absolutely. So my name's Harrison. That's Harrison. No, so I'm gonna be, I guess, a fourth-year PhD student now at the University of Georgia, studying respiratory syncytial virus and vaccines against it. So I'm more in the viral immunology camp.
Just the right paper for discussion.
Mm-hmm.
Samriti?
I'm Samriti. I am a fifth-year PhD student at the University of Ottawa, and I work in a cancer immunotherapy lab, and we use oncolytic viruses and antibodies to make prime boost vaccines.
Cool stuff. Eugenio?
So I'm a second-year PhD student, and I'm working at the National Autonomous University of Mexico, and I'm working with Tregs and TGF beta signaling pathway.
Oh,
Very close to Jatin.
yeah. Tanu?
I am Tanu, and I'm a fourth-year PhD student in University of Florida. I work with norovirus and microbiome and how their interaction affects the host immunology.
Natalia?
Hi, I'm Natalie, and I'm working at City of Hope, which is a comprehensive cancer center in Duarte, California. So it's like 7 a.m. for me. But I work on microRNA, specifically in the context of the development of different immune cells. So I'm a fourth-year PhD student, so working on like B cells, but I just did a Treg paper as well. So.
Yeah, we got very little diversity when it comes to people working on Tregs.
Yeah.
All right, and I don't know why I keep calling it Natalie, right? Natalie.
Yeah, it's Natalie.
Yeah,
Thank
my brain does very well when I have to switch between English and Spanish names. and there's just one exception of Harrison. That's the one name that I don't know, just doesn't work for me. Harrison.
It's all good, man. We're good.
All right, let's begin.
And Maria.
Maria, yes. Oh, my stupid Zoom window. It did not
That's fine.
show. It did not show the last name for me.
So I just graduated from University of Warsaw. I was working on adipose derived stem cells and how they affect immunity. And I'm starting PhD in October, and I will be examining the effects of changes in glycosylation on some parts of immunity, I guess. I don't know the details yet.
That's pretty cool stuff. I have a feeling after the Treg discussion in the next podcast, we will pick up something for stem cells. That'll be cool. Okay, Harrison, stage is all yours.
Excellent. Okay, so guys, thank you so much for joining us. To everybody on live stream and everybody watching this recorded, thank you so much for tuning in. For those of you who are in our Zoom right now, when you have a question, if you have something to add, if you have an anecdote, go ahead and immediately throw it in. If you end up waiting, you'll probably forget it. So, questions, comments, anything, just throw them out there. So today we are looking at a paper from Journal of Immunity called Acute SARS CoV2 Infection Impairs Dendritic Cell and T Cell Responses. So I chose this paper because obviously we cannot escape getting around coronavirus. It's everywhere. You have to wear your mask to the grocery store. I figured that it was appropriate that we do Journal Club using SARS COVID as the model. Moreover, I think dendritic cells kind of get forgotten as far as their implication in protection from disease. They are our professional APCs. And then T cells are also, we were discussing this prior to the stream, that T cells are so important fighting viruses. And when we're looking at a vaccine, our primary endpoint is looking at B cells and antibodies, right? But we can get antibody responses in some things, but they're not protective. And so I think that most of the time, it's because we have a poor understanding of how the T cell responses. So I thought that this paper was good to bridge innate and adaptive immunity. And then we're also able to look at actual patients. So we have a very appropriate model here. I thought it was really, really cool. And it's all happening in real time. So very appropriate. So with that, I'm going to go ahead and get started just outlining what we're going to talk about for the background. I decided against talking too much about actual COVID-19. I think that everybody is very familiar with COVID-19 and all the issues around it. So if you're not, I suggest turning on your TV and just checking out what's going on in the world. So we'll skip that and we'll focus mostly on the virology of SARS CoV2. I'll probably end up calling it SARS or COVID or something like that, but it always just refers to the SARS CoV2 and the COVID-19 disease that it is responsible for. Also briefly discuss T cells, their relationship with B cells and DCs, and then talk about the difference between the DCs. And then prior to figures, if there's background that's appropriate, I'll present a little bit of that background. So first, I want to start with the virology aspect of it. So we've all seen the picture behind me. This is the coronavirus picture that's on your nightly news. And then the reason that we call them coronaviruses is because under EM, they look like a crown, corona crown. And the reason that they do is because of their spike glycoprotein. And so this is a main immunodominant antigenic surface protein. We also have the other structural proteins that I'll talk about here in a minute, but the order is nidovirales, family coronavirus, and then within the coronaviridae, there's 4 genus of coronaviruses, and we're talking here about the beta coronavirus. These are associated with zoonotic infections. So the genome is a positive sense single-stranded RNA, which means that its genome is mRNA. So it can go straight into translation. And that has implications as far as the ability to produce proteins and how quickly we're able to see viral translation. The genome's around 30 kbs. And so if we look here at the schematic of the genome, we know a lot of open reading frames. Some of these open reading frames have function. Some of them are unknown at the time. And so I'm just going to focus on the structural proteins when introducing coronavirus. So like I mentioned, there's the spike protein. And so this is responsible for attachment. So it's essential for virus to enter host cells. So it has an affinity for the ACE2, and this is a receptor found on host cells. And so when the spike protein attaches, another host protein called TMPRSS2 will cleave the spike protein, which then, once you cleave the spike protein, you have 2 subunits, S1 and S2, and S2 is responsible for the actual fusion of the virus, and it contains what's called the RBD, or the receptor binding domain. The other structural proteins are the membrane, M protein, and so this is responsible for budding. So if we think about it like influenza, spike is kind of like the HA and membrane is kind of like the NA. There's also the envelope protein, and this is responsible for things in the virus life cycle, assembly budding, envelope formation. And also we're noticing that it's related to pathogenesis in the cytokine storm and inflammasome activation. And then there's the nucleocapsid protein, which is associated with the RNA genome. So over here is just a quick, easy to see that we're getting this ACE2 binding, the TMPRSS2, the host protein is cleaving, and then we get this S2 that's able to infect host cell.
I have a question. Can you go back
Yeah.
to the previous slide?
Sure.
Actually, I have two questions. So just as having zero knowledge about viruses, do you know if positive stranded viruses can they start translation faster than negative stranded viruses?
Yes, and so the reason that is, and granted that might not have any relevance in real time, but so positive sense means that we're looking at a 5' to 3', and so when you get you're able to get translation immediately, whereas the negative sense single-stranded viruses such as respiratory syncytial virus requires an RDRP, which goes from 3' to 5', and then the RDRP changes the genome to 5' to 3' before you can get translation.
But even positive stranded viruses need RDRP.
Right, they need RDRP to replicate, right?
Okay.
But not to translate into protein. So they can take, before they even worry about replication, they can immediately start translating protein. And they will use the RDRP, of course, to replicate the genome for future virions.
Okay, are you
Just
saying
to-
if the virus does not have RDRP, it will still infect?
Well, it can still infect in that if you have your spike glycoprotein, but if you don't have RDRP, then you're not going to get replication of the genome.
So basically, the cell will be susceptible to the virus. It can enter, but it is not permissive because it cannot replicate and produce more.
Exactly.
And also for almost all the viruses, translation always happens before replication. So you cannot do replication without having the proteins, which are important for replication, which is produced during the translation process.
There's kind of a gradient that happens where they'll focus on their translation, and then when they hit this peak that's different virus to virus, they'll then switch their function into more focused on replication than translation. So that's actually really interesting, and more on the permissiveness. So the virus that they look at here when they're looking at their neutralization assays, they're using a pseudotype virus. And so you can use pseudotype viruses when you're concerned about attachment and things like that, where you don't really care about permissiveness. Now, granted, they do here, but still, if you have a viral vector and you were to use, say, VSV as your viral vector and you take the spike protein, you're able to get infection, but you're not able to get it's not permissive, so you're not going to get continuous copies of it. So that's useful in the context of creating protein vaccines.
And the last thing, do you know if this TMP-RSS, is this also required for just general ACE2 signaling, or is it something completely random out there?
Is what, sorry?
The TMP?
Oh, TMPRSS2? Yeah, so TMPRSS2 is just another host protein that's found. And it would be required because that's going to be that's what's going to cleave the spike protein into the post-fusion. Where you get the S1 and S2 epitopes, and the S2 is a conformational, needs to be in the right conformation in order for you to get fusion. So you can't really get entrance if you don't have TMPRSS2. But TMPRSS2 and ACE2 are both found abundantly in host cell.
Okay.
And is this a unique requirement of COVID-19, or is this required for all coronaviruses? Do you know?
So I think that's a good question. I don't know. I know that TMPRSS2 is also implied in other infections such as influenza. But as far as the spike protein, I don't know if if it's required to have TMPRSS2. So some of the differences in the pathology of coronaviruses is we're looking at if they stay in the upper or the lower respiratory tract, if they have affinity for receptors on the lower respiratory tract. And I don't know if the spike protein one is similar enough to, the standard coronaviruses that cause a cold, that it has the same kind of fusion requirements. And then I also don't know exactly where TMPRSS2 is spatially in the body. So if there are certain cells that have more TMPRSS2, so I'm not sure if anybody has any comments there.
I think it's in the airway epithelial cells, both the ACE2 as well as the.
Right. So they're both found in the airway epithelial cells. I don't know if TMPRSS2 is found anywhere else.
Yeah, no idea about that.
Yeah, not sure. I should, but I don't. Yeah. Is that cool?
Yep. Yeah, there's so
Right.
much happening all around. It's very difficult to know everything. It's
Oh yeah.
yeah.
Oh, I know. Yeah. So here we're looking at a cartoon of what SARS infection looks like with our DC and our T cells. So we know that dendritic cells are your professional APCs, your antigen presenting cells. So if we look here, we have a nice infection. We get replications where the virus is moving down to the lower respiratory tract, where we're getting endocytosis, antigen presentation, dendritic cells, and then it's going to show your CD4 and your CD8 T cells. So we'll start here in the CD4 cells, where they're going to show the CD4 cells. You'll get clonal expansion of these T helper cells, which will prime B cells, as well as help with CD8 T cell priming. And then your B cell priming will result in antibody secretion. And of course, antibodies will help neutralize virus, and then also help bring out other inflammatory cytokines. And then the dendritic, sorry about that. The dendritic cells also present to CD8 T cells, and these are your CTLs, your cytotoxic T lymphocytes, which then result in clonal expansion, which ideally results in viral clearance and inflammatory cytokines. And then within the CD8 T cells, there's some variety of function here where they'll directly kill apoptotic cells using perforin and granzyme B. And then there's also the upregulation of Fas ligand and TRAIL, which is going to lead to apoptosis. And then, of course, you're going to get your pro-inflammatory cytokines.
Harrison, can you explain what is cross presentation? There'll be
Cross-
something very important here in the dendritic cell.
Cross presentation?
Yes.
Not sure if I know what you're.
Oh, I mean, it's something that I learned, I think 2 years back, and I did not know something like this happened. I think
talking about MHC 1 class 2?
Yeah, so because dendritic cells, they take up stuff from around them through the phagocytosis pathway or just endocytosis pathway, and naturally such a thing should be presented only on MHC2, but through cross presentation they manipulate the pathway so that even something that was endocytosed gets presented on the MHC1, so cytotoxic T cells or CD8 positive T cells can also detect and get activated.
Oh, okay.
Yeah, this is a very important part to show something on an MHC1 without actually being infected by that thing.
Oh, sure. Yeah. Do you want to talk a little bit briefly about kind of the differences between MHC class 1 and 2?
Oh yeah, sure. Also, if anybody else wants to take it up, that's also fine. Else, yeah, of course, I can go ahead.
Yeah, go for it. Why don't you talk? Just give us the difference between what we're looking at with your MHC 1 and 2 in the context of maybe the length of the peptide that it's presenting. That sort of.
Okay, so dendritic cells, like some other antigen presenting cells, these can present their peptides in two ways. One of them would be called the class 1 MHC. Class 1 MHC is present not only on dendritic cells, but every nucleated cell. And a class 1 MHC will show peptides. These are shorter peptides, around 8 to 11 at best, peptide lengths, and these will present things that the cell itself is producing. So I think of these MHC1 like ID cards. You're showing your ID card everywhere. You're going around telling this is something that belongs to me. And CD8 positive T cells, they check your ID card and the contents of the ID card. They tell you if something you are presenting is foreign or is yours. And if it's foreign, you get decimated. If it's not, you are allowed to live. MHC2 is only restricted to cells that are presenting antigens. That is, when they take up antigen from outside, it could be through just eating it up as phagocytosis, or it could be through some form of pinocytosis or endocytosis, where they take in stuff from outside and they present it on another displaying molecule called MHC2. This is supposed to show the CD4 positive T cells that it was picked up from the environment, it does not belong to me. And that item that is presented on MHC2 is looked up by the CD4 positive T cell, which is a helper T cell. If it can recognize it, it will license this dendritic cell for cross presentation. That means now this dendritic cell knows something's up with whatever I picked up. There was a helper T cell that was specific for this, and this licensed dendritic cell will take that materials from the MHC2 and it will now present that on MHC1, so it can activate the cytotoxic or CD8 positive T cells, because in the end, helper T cells are just helping. They're not killing things. The cytotoxic T cells, they're going to go cell to cell and kill virally infected cells. So for this, understanding the two class MHCs is very important. Anybody who's watching, please take notes.
Yeah, that was great. Yeah.
And yeah, as you said, one more thing, that MHC class 2 peptides, they are slightly longer, could be around 15 amino acids.
Yeah, the elementary saying of the difference between the CD4 and the CD8 as far as the peptide goes. So CD8 is more like your hamburger. So if you think of the patty of the hamburger as the peptide sequence, and then the bun is the MHC class 1, where you're going to get the entirety of the peptide inside of that. The whole epitope is inside of the MHC class 1 molecule. But the CD4 is sometimes like a hot dog, where the wiener of the hot dog is your peptide sequence. It doesn't always fit in the bun of the of the MHC class 2. So that can have different implications in immunology. I was reading a paper once about bat immunology, and I don't remember the specifics, but it's something about the way that they're able to present their class 1s, that they don't really get viral infections, right? Because bats are a host of a ton of infections, but they don't really succumb to a whole lot of viral infections. So interesting little side.
You know, when I read that in a book, I think it was QB, when they made this hamburger and the wiener analogy, and I knew right away that this book is written by an American.
Yeah. Well, you got to put stuff in words that
context.
We all yeah, hot dogs and hamburgers. All righty. So then I want to look a little bit briefly, and I'm sure we'll get some comments here, hopefully from everybody, talking about activation and class switching of B cells. So I'll just follow this cartoon all the way. So your antigen presenting cells presents antigen to your T helper cells, which we talked about just now, where we have the MHC class 2, and then your TCR. You have these co-stimulatory molecules, B7 and CD28, which activates your helper T cells. So then we have our activated T helper cells right here. They interact with the B cells with the CD40 and the CD40L. This activates the B cells to proliferate, differentiate, and secrete antibodies. And then your T helper cells are also secreting cytokines that determine class switching. So IL 4, 5, 6 is your Th2 cytokines, and then interferon gamma, I think IL-2 class 1 are your Th1 cells. It's been a minute since I looked over that, but it sounds about right.
Yeah, interferon gamma is for...
Yeah, am I missing one for the...
TNF alpha.
TNF alpha, thank you. Okay.
Hold on, are you saying TNF alpha helps in class switching?
No, it's just a Th1.
Okay. You're just
Cytokine.
naming Th1 cytokines. Okay.
Yeah. Okay. So then we have, our B cell and your different classes of antibodies, so IgM, IgG, IgG, IgA, and then there's IgD, nobody knows what IgD does, right? Are we still there where we don't know what IgD does?
I think
we
at this
don't-
point we don't care anymore.
Yeah.
about it.
Yeah. So briefly about the classes, won't spend too much time. So IgM is going to be the first thing that you're going to get. It's got 5 of these IgGs attached to it. Your IgE is the allergy associated immunoglobulin, IG, what's it called? IG. IgG is like the super important IgG that we look at. And then IgA is the mucosal immunity. Anybody have anything that they want to add to expand on B cells and class switching, anything like that?
I guess it's important to talk about like there's somatic hypermutation that's happening during this reaction. Like there's basically this whole evolution that is happening within the germinal center, which is the mini structure where this basically happens. Except there is an extrafollicular reaction, but I'm sure that's not related to this. But under somatic hypermutation, it's just randomly mutating, and then you'll go to the next phase where after it's proliferated massively, it'll get selected out the ones that bind the best, and then you get to switch into whatever class switched antibody this guy's going to be making for the rest of its little life. So that's also a DNA rearrangement step. So it's like it has to be really tightly regulated because there's like the proliferation steps and these DNA regulation steps. So it's really kind of a scary thing that can happen.
Yeah. And just on what Natalie was talking about for extrafollicular, I think that there is actually something extrafollicular going on even in this paper, even though they don't talk about it. And when they talk about that discrepancy in the T cell response and B cell response, maybe we can talk about it when we come to that part of the figures.
I usually can tell because, I mean, a lot of the extrafollicular stuff, it's just IgM.
Yeah, and it's not dependent on T cells.
Yeah.
Which is why they're the first Ig response that you're gonna
Right.
get.
And if you think about it, actually these extrafollicular B cells that are making antibodies without the help of T cells, these antibodies are helping in opsonization of antigens, which then get taken up by antigen presenting cells, and then they are presenting these antigens. So if you look at it, the extrafolliculars might be the first responders.
Yeah, it really highlights the fact that our immune system is this constant feedback loop.
Yeah.
Is exhausting for graduate students and the body alike, where we have this constant, I mean, it's crazy. Like and immunology is still such a young field that we're still learning about all these classes of different cells and cytokines and things like that. It's crazy.
It's very non-welcoming to outsiders. People get afraid looking at these weird numbers.
Yeah, well, so do I. So,
I thought you were talking about the immune system was unfriendly to outsiders, not just immunology as a field.
so philosophical, right?
I guess it depends on if you have an autoimmune disease, if you like your immune system or not, right? Or if you have an allergy to peanuts.
Yeah.
All right. So moving on to that, I just want to briefly touch about the difference between the two types. The two types of dendritic cells. So we have our conventional and our plasmacytoid immune cells. And so what I really wanted to highlight, and don't want to spend too much time on this because this is an exhaustive cartoon, but also what I want to show is with the PDCs, something that's going to be super important to us is going to be type 1 interferons. So there are different classes of interferon, and everyone's pretty familiar with interferon gamma. It's a type 2 interferon. But interferon alpha and interferon beta are type 1 interferons, and these are extremely important for an antiviral response. And these are something that I think get overlooked pretty often. So it was pretty cool to see a paper kind of focusing on the role that PDCs have in interferon type 1 interferons. If there's anything inside of this exhaustive diagram that someone wants to talk about in detail, we can go ahead and do that.
This weird thing that I see NF-kappa B coming outside the cell, or are they?
I don't think NF-kappa B is coming outside of the cell. I think that the Earth
they ran out of space to draw it inside.
Yeah, exactly. Yeah, not to scale. I don't think your dendritic cells are visible on a PowerPoint slide. So forgive them.
Can you just in one line, if you could differentiate between conventional plasmacytoid, what would it be? I honestly don't know much about plasmacytoid. I just, yeah, I don't hear about them that much.
Yeah, so in this paper, let's just say type 1 interferons.
Okay.
I mean, there's a lot of differences as far as their maturation process, their presentation, the involvement with different cytokines and stuff. But for this paper, let's just focus on interferon alpha, interferon beta.
And how much do plasmacytoid DCs make up in terms of percentage in PBMCs? Do you know?
That's a good question. I have a background on PBMCs coming up, but I don't remember.
Like, would it be very difficult to isolate and study them from human blood?
No, so the authors use a pan DC isolation kit, and you could just use beads and isolate them out of blood. And I'm pretty sure that's exactly what the authors did here when they're isolating their PDCs.
These cells don't even look like dendritic cells.
Yeah,
and they probably even have a different development pathway.
Mm-hmm. Yeah, they definitely do. And looking at this paper, I should have done more background on the difference between them, but there is a lot of stuff to get in. I do know that a lab next door to ours does a decent amount of work on the difference between the two dendritic cells. So I know that they're extremely important for pathogen protection. So, but a quick Google search, I didn't find immediately the percentage of PDCs and PBMCs. So all of DCs in, I guess, PBMCs, we're looking at 0.2 to 1% of PBMCs. And I don't know what percentage of natural conventional versus plasma. But I do know that the paper will talk about a potential biomarker for severe disease based on the ratio of your two DCs. So a quick overview on the methods. So they're looking at human subjects, which is really, really cool. We're looking at patients that have an acute infection or a convalescent compared to healthy donors. They use a decent amount of flow cytometry where they're looking at different panels using 12-color analysis. They're looking at secreted cytokines by a 13-plex panel. They're doing their intracellular cytokine staining after stimulation of cells with PMA ionomycin, and then using like Golgi plug in order to retain those cytokines for flow. So back to the DC, looking at DCs. So yeah, they isolated it by a pan DC enrichment kit. So I imagine that if you go to Thermo and you ask for a pan DC enrichment kit, that they'll sell it to you. I don't exactly know how that works, but I imagine it's some kind of bead-based isolation method. And then they would stimulate these with a pro-inflammatory cytokine cocktail. They wouldn't use PMA ionomycin. It was like a mix between like TNF alpha, IL-2. Yeah.
Was it 6 and 1 beta?
The cocktail?
Yeah, maybe I could be wrong.
So for the DC cocktail, IL-1 beta, IL-6, TNF alpha.
Okay.
Yeah. And then prostaglandin E2. So I don't know if that's the same thing as estradiol. Is that estradiol?
I don't think so, but could be wrong.
No, it's PGE2. Yeah.
PGE2 is, yeah, it's some lipid mediator, but yeah, not sure.
Gotcha. I got hormones on the mind. They also do a mixed lymphocyte reaction assay where they take these DCs and stimulate them, and then they gamma irradiate to prevent replication. They isolate the T cells from this using a pan T cell isolation kit and then stain with a proliferation marker, CFSE. So then they mix these together with the DCs and the T cells from healthy donors where they're looking at the function of the DCs, and then they'll measure the proliferation after 5 days.
And how many days after infection did they say that a patient was acute or convalescent?
I think it was 30, where they considered them convalescent. I mean, it should be at least 4 weeks. I want to say somewhere in the paper they mentioned that they recruit patients from the acute into convalescent after like 30 days.
Yeah, I couldn't find that info either in the paper.
Okay. Maybe it's in there, maybe it's not.
Oh, I think I see in figure 1. 17 acute patients, 25 convalescent patients were collected a median of 13 and 30 days after symptom onset.
Oh,
So
Among-
yeah, 2 weeks and 4 weeks.
okay.
Okay, yeah. So the methods state that among the convalescent patients who received treatment, subsequently discharged, 29 blood samples from 3 severe and 26 mild patients were collected at the median 30 days after symptoms onset. So 21 to 54 days after they were symptomatic is when they were considered convalescent. And then they do LE spot with the PBMCs, and then they're looking at a peptide, either the RBD peptides or NP protein, and then addition of stimulators, the anti-CD28 and anti-CD49.
What's the 49 for?
I mean, I think it's a stimulation of the.
I gotta read on that. I've never heard about 4090.
It's an antibody against it, and CD49D participates in mononuclear cell trafficking to endothelial sites of inflammation, has a role in cell-to-cell interaction and cell adhesion to extracellular.
Okay.
Yeah. And then they looked at interferon gamma. Then they did a neutralization assay using a pseudotype virus where they had their heat inactivated patient serum plus pseudovirus with a luciferase reporter in these ACE2, these HEC293 cells expressing ACE2. So they're able to use this as the pseudotype viral newt assay. Then they're looking at the luciferase after 48 hours. They also did a pretty standard ELISA and qPCR, where they were looking at with the RBD bound to the plate, and then serial diluted plasma, and then HRP and TMB, and then their PCR came from saliva from RNA extracted from saliva. And so with that, we'll go ahead and jump right into the paper. So they use throughout these definitions AP, CP, and HD. Instead of, I guess they use healthy donors instead of healthy patients. But so they have their acute patients, and they define a difference between the severe and the mild based on the need for oxygen supplementation. And so table 1, what we're looking at is a clinical characteristics of the patients in the study. And what they noted as significant is that your severe patients have chronic comorbidities more significantly than your mild patients. You also have a reduction in your lymphocyte count for the severe patients, and then a reduced platelet count. So this was the first data that they saw, and they thought that you're getting this lymphocytopenia where you have a reduction in the lymphocytes, and that could result in an increased disease. So yeah, here's that slide on PBMCs that I was mentioning, because I also wanted to talk about myeloid deprived suppressor cells, since the rest of the cells that they talk about are pretty standard. MDSCs are not as often talked about. So they get their PBMCs after just centrifuging blood. So you would use some kind of a gradient filter, adding in, add blood to that, and then you centrifuge it. And that separates based on density of the plasma, PBMCs, your PMNs, and then your red blood cells. And then below is just a a general frequency of what's found in the PBMCs. So our dendritic cells are looking, 1 to 2% of those PBMCs are dendritic cells. And then I also wanted to talk about these myeloid derived suppressor cells, because these will become important in the next figure. So what's important to note here is that these are like anti-macrophages, so they reduce APC sensitivity. There's a lot of downregulation in that they reduce proliferation and activation of your T cells and your NK cells, so that's going to be important. And so showing that in this figure, so what we're looking at, so figure 1's acute infection results and broad immune cell suppression. So we looked at that table and we saw that they had a reduced platelets, and so what we're looking at here is flow on those PBMCs. And so we're looking at, starting with A, we have our T cells, our B cells, our NK cells, and the black circles, your healthy donor, your blue squares, your convalescent patient, your purple triangles, your acute patient. And so what we notice is that we have a significant reduction in our T cells in our acute patients compared to our healthy donors, as well as our NK cells. However, B cells, there's no significant difference there. And then if we look at our innate cells, there's a significant reduction in your healthy donors and DCs for not just your acute patients, but also the convalescent patients. You have a reduction in monocytes in your healthy donors compared to convalescent and acute patients. And then what was particularly interesting is you get a significant increase in your MDCSs in your acute and your convalescent patients. So what we see is the increase compared to the healthy donors, and basically what this is showing is that the infection reduces T cells, B cells, NK cells, DCs and monocytes, even after infection, so your convalescent patients, and then your suppressor cells are increased. So this really goes to show what exactly is happening in the population of the reduced platelets and the immune cells that they noted in table 1.
I found it very counterintuitive, because all this time I was told that in COVID-19, it's the T cells that are wreaking havoc. They're going out of control and causing immunopathology. But here it looks like they're going down.
Mm-hmm. Yeah, it is a little bit counterintuitive, because when you hear what patients are dying of, they say cytokine storm, right? And so when you think of cytokine storm, you'd think of an upregulation of pretty much all your immune cells, right? Because you would want, in order to get cytokine storm, you'd want a lot of immune cells in order to create that cytokine storm.
Yeah.
So I thought that was super interesting.
It would also be cool to check these patients again, maybe after 3 months or 6 months, and see if they ever get back to normal levels.
Yeah, for sure. Is there long-term immunosuppression here?
Mm-hmm.
And then is that implicated in any protection or even enhanced disease the second time you get it? Would also be some of the implications there. I thought that was really, really neat. And the cool part about it being a cell press paper is that they have these beautiful cartoons that will summarize exactly what pretty much each figure is. So just wanted to touch briefly on how the DCs and the T cells interact, especially when we're looking at these different ligands and receptors. So I don't need to just repeat this, but this is something that's going to be important here for the next slide. That there are a decent amount of interactions that occur between the DCs and the T cells in order to have T cell function. DCs need to be able to tell the T cells what to do, and it's done so through these different interactions here noted in the middle. So figure 2, we're looking at DCs derived from acute patients, and we're seeing a reduced maturation potential. So we're looking here in figure A at the total number of DCs. Again, we have our healthy donors, our convalescent patients, and our acute patients. And so we don't have a significant difference between the percentage of PDCs between patients. However, when we look at it in the context of a ratio between your CDCs and your PDCs, that we have this bias towards way more CDCs for your acute and your convalescent patients compared to healthy donors. So this is really cool because it's got the potential for a biomarker for showing disease. So I thought that was pretty cool.
Now, is this something that's very common to do, to look at the ratio between CDC and PDC?
That's a good question, and I think so. I would think that they were basing this based on some idea that the ratio is significant between them. But then again, the PDCs are a relatively new class of cells described. I don't remember exactly when, but it's relatively recently that the PDCs were recognized and that people are starting to study them more. So actually, I don't know.
In the previous figure, did they say that the DCs were going down? And like as percentage of PBMCs, the DCs are going down, but as a percentage of DCs, CDCs are going up.
Yes.
Okay.
So
but the level of production of interferon, type 1 interferon, would still kind of, I would assume, be the same if you're having the same percentage of PDCs regardless of the ratio. So that means that I would interpret that to mean that there's some other immunosuppressive mechanisms that are somehow suppressing that type 1 interferon. And that's where the MDSCs would come into play.
Exactly. Yeah, that makes a lot of sense. So I think I got ahead of myself when I talked about the biomarker. I got really excited about that. So we'll keep going on with the figure where we're looking at the different markers for the CDCs. And we see a significant increase of CD86, and that's a co-stimulatory marker. So that's going to be important when we're looking at the T cell function here in figure A. And then figure B, we're looking at stimulated functional assessment. So this is the PMA ionomyosin stimulation, and we see reduced maturation in your AP and your CPs, and this is noted with the CD80, CD86. And then C continued? No. I guess this is this? Oh, C's here. Okay, there we go. Sorry. So yeah, so this is still B, where we have a significant decrease in stimulated cells. So the cells, even though the percentage is the same, functionally something's going on here, as you know, as you stated, that when we stimulate these dendritic cells, that we're not getting type 1 interferons. And then if we look at C, we note also a reduction in CD4 and CD8 cells in your acute patients. So, I mean, this makes sense. And that's measured using CFSE low percentage, so proliferation marker. So the conclusions here is that SARS infection reduces PDC maturation and functionality, particularly the type 1 interferon, and then responsible or related to the T cell lymphocytopenia. Moving on to figure 3, we're looking at peripheral T cells displaying a loss of a functional loss during infection. So this Ki67 positive is a cell proliferation marker. CD38 HLA-DR is a T lymphocytopenia marker. And then PD1 is our T cell exhaustion marker. And so going through looking at our percent positive, we have higher proliferation in our healthy donors compared to our acute and our convalescent patients.
I have a question.
Yeah.
Anybody knows how Ki67 works? What does it bind to?
So I saw that it was related to cancer biology and about proliferation, but that's all that I looked into. Marker for proliferation. Okay, during interphase, Ki67 antigen can be exclusively detected within the cell nucleus, whereas in mitosis, most of the protein is relocated to surface chromosome.
Oh, so wait, what are we measuring on the surface? Or did you say it's relocated in the cytoplasm before that?
Within the cell nucleus
Okay.
during interphase, and then during active phase of cell cycles, and then absent in resting cells. So I guess it's only around during proliferation.
Okay, so this looks like an intracellular stain.
Yeah.
For the okay, I thought it's just some dye that binds to DNA, but it looks like it's a legit protein.
Yeah, it must be, right?
Yeah.
So they're basically just showing the loss of function of our T cells with different markers here. And then here's our CFSE low. So this is a proliferation marker so CFSE likes to stay within cells, and it can stay in for a handful of generations. However, it eventually loses staining. And so we can look at percent proliferation based on what percentage of your cells have a low stain with CFSE. So it's inversely correlated with proliferation, which is why we're looking at high or low percentage CFSE is high proliferation. So they took the CD cells and proliferated with stimulation by CD3 anti-CD3 anti-CD28, and we can see using the CFSE that we have a reduction in proliferation in our acute patients, which is what we expected to see. And then looking functionally at our T cells, we have a reduction in interferon and IL-2, but there's no significant difference in TNF alpha.
I just want to add on to the CFSC thing. It's like you stain the first generation of the T cells with the CFSC, which is like in the DNA.
Mhm.
So each time that there's a
division.
Yeah, division. I'm sorry, it's so early.
Yeah,
each time there's a division, it's actually getting diluted, and that's why you're supposed to see like 5 peaks. But you can see in the purple, you can see that it's just kind of like one monster peak that's not, doing the proper thing.
Gotcha.
I'll just make one correction to that. So it actually binds to the lysines on the proteins.
Oh, good catch.
Yeah. So every time the cell divides, those lysines, like proteins, also divide, and the stain gets diluted. So the principle is the same.
So I have a question regarding this figure, and I want to hear your point of view. In figure B, like they took out PBMCs and they plated with CD3 and CD28, and they look for proliferation. But you start with a population of having a lot of myeloid-derived suppressor cells. So maybe the results are just because you have like a more immunosuppressed environment. So I would expect to do this type of experiment by purification only CD4 cells or CD8, and then plated bound with CD3 and CD28 in an environment without this anti-inflammatory response to somehow conclude that the phenotype is intrinsic to the cell and not extrinsic to the environment of the patient.
Mm-hmm. Yeah. Wait. Did they take PBMCs for B?
So PBMCs, they didn't purify CD4 or CD8.
Oh, okay. Yeah, you're right.
And I didn't like the conclusion of this figure because it's somehow biased to the microenvironment that you have.
Right.
No, that was a good catch. Yeah, I missed that in the methods. When I was looking at the T cell proliferation, I noticed that they had used a pan T cell isolation kit, but that was for the MLR, not for this T cell proliferation assay. Where you're totally right, they did label PBMCs, and is CFSE wouldn't be specific to T cells.
No.
So yeah. So I mean, I guess they would just use some panel where they would be able to isolate it, by gating the CD4, CD8s. But no, that's still interesting that they decided to look at the whole PBMC and not isolate T cells. That's a good catch.
Yeah, and as
well, I mean, I guess with that type of
oh no, Natalie, you go.
I guess with that type of proliferation, like those would be the cells that would survive and live, right? So presumably everybody else would die.
You mean when you're giving anti-CD3 and 28 only T cells would
Yeah,
survive?
I don't know, but like, would that happen? Like that would only stimulate the T cells to survive past that point?
Oh, I think that's for sure. But what Eugenio means is there are still, let's say, MDSCs around, so they could be secreting factors that are suppressing these T
Yeah,
cells.
sure. I agree. Yeah.
I think what's the proliferation? The proliferation does exactly say what the T cells are proliferating, because they probably did gate on that, but it doesn't mean that the environment did not have other non-T cells. So
Exactly.
they did have an effect.
Yeah. But that, I mean, would that, I mean, yeah, it'd be interesting to look at the T cells that are isolated. But at the very least, it gives them more credibility to their, the significant change in the MDCSs, that those are having a functional role. So even though they're still in the PBMCs, we're able to see that stimulation results in reduction of proliferation because of these specific MDCSs.
Yeah, that is true. Although you can't say that there is something intrinsically wrong with these T cells, but as the sum of everything going around them.
Mm-hmm. Yeah, no, that's a really good point and a really good catch. I appreciate that. Any other questions? All right, so continuing on figure 3. So we're looking at stimulated T cells, and then we're looking at the different T cell populations, our central memory or our effector memory, RA positive. And so when we look, excuse me. So here at the top of C, we're looking at our CD4 versus our CD8s and pulling out our functional cells, our INF gamma positive and TNF alpha positive cells. And then we're looking at what population of these are our functional cells. And so as we expected for interferon and TNF alpha, the central memory and the effector memory of our CD4 cells have a reduced function. For our CD8 cells, we also have a reduced function. So then if we take this effector memory RA positive CD8T subset of cells where we have a reduced function, we then look at that in figure D, where we're gating CD8 T cells based on the different markers of their subclasses are again our effector memory RA, central memory RA naive, and then looking at these in functional assays where we're looking for positive granzyme and perforin, and these are non-stimulated CD8 cells, and so we have a reduction in functionality through granzyme B and perforin, and then a reduced T cell count as well.
Anybody wants to say what's the difference between EM and MRA?
Yeah, someone please.
All right, I'll just say it then. So if you in is it starting from naive T cells to memory T cells, it's a spectrum. So you have the least activated, naive, and you have the most activated, which require minimum stimulation, that is the memory. And between those, we have made these intermediate types. So EM is, and so they have mentioned in this, if you look at the figure D, that flowchart. So naive, if you look at naive, that's the least activated. Then there are central memory. Central memory T cells they have been activated once, but they still require co-stimulation, and they still need to go inside the lymph nodes or spleen or secondary lymphoid organs to get activated again. So that's why they express CCR7, which is a chemokine receptor that lets them inside the secondary lymphoid organs. Then EM is a next stage of activation. These cells are poised for activation. They just need a stimulation. They don't need a very strong stimulation, but they need little stimulation. That's why they don't need to be inside the secondary lymphoid organs, and they lose the necessity to express CCR7. And the closest to the memory, true memory phenotype is the EMRA. these express CD45RA, and they don't even express CD28. That means they don't need co-stimulation at all. They just need some TCR stimulation, and they're good to go. If I remember correctly, this CD45RA, because do you guys know where CD45 comes in the picture when you look at TCR signaling? It's a phosphatase, and it dephosphorylates some proteins in the TCR signaling pathway. So this CD45RA, it's placed in a way that it does the job better of dephosphorylating proteins than other CD45 isoforms. And that's why these T cells get activated fairly quickly compared to others. So if you look at just the y-axis in the granzyme and perforin, you'll see that all the way the MRAs are producing more granzyme, or they're more percentage of MRAs that are producing granzyme compared to the EM. It's just because they don't require as much stimulation as the EM. So look at all of this as everything falling in a spectrum.
So when I've looked at T cell markers before, and there's CD45RO, is that right?
Yes, so I think RO is even, I think it's even closer to a memory phenotype, because if, as long as I can remember, the 45s, they are placed slightly different compared, they localize on a different place with respect to the TCR on the cell membrane, and based on their either farther localization or closer localization, they either work better or worse, or they either amplify the TCR signaling more or less. I'm not sure which way works better, like if they're farther, if they're working better, but that's somewhat the known concept about it.
Gotcha. And then is this CD45 different from the CD45 marker that we have on like our innate immune cells if you're doing immunophenotyping on flow?
If I remember, all is it all leukocytes, or probably all leukocytes express CD45.
Okay. And then
this is an isoform that is specialized for
the T
probably
cells,
these T cells, RA.
Okay.
There's
Gotcha.
also, so the CD45 has so many isoforms. It's so confusing for newcomers in this field, because B cells have their own CD45 isoform, which is called B220.
B220, yeah.
Yeah.
So when I was doing my first flow panel, I had everything set for my innate phenotyping, and I had CD45RA instead of CD45. And I didn't realize there was a difference until like 3 weeks before I had to run flow. So
it's so confusing.
Yeah. But I appreciate that very concise, easy to understand explanation. That was really, really cool. All right. Figure 4, impact of disease severity on our AP-derived immune cells. So we're looking at our acute patients where they divided them into the mild and severe. Mild, we're looking at purple. Severe, we're looking at black. So going off of what we would expect to see based on the data presented in the other figures, there's a significant difference between the severe versus the mild, where the severe patients have a significantly increased amount of MDCSs. And then here we're looking at the biomarker that I jumped way ahead of myself, where we're looking at the ratio here between the severe and the mild patients, where if you are infected and you have a severe infection, you'll probably have a significant increase in your CDC to PDC ratio. Whereas if we're looking at just infection in general, even though there's like a couple of patients here that have a high number of PDCs in mild, there's no significant difference anywhere else except the places noted here. So that could be a really cool biomarker for determining disease outcome. And they mention a ratio of 50. So it looks like, based on not looking at healthy donors, but just these, that your percentage of CDCs and your PDCs may not change all that much, but the ratio may be significantly different. Moving on to figure 5, where they crammed a whole lot of stuff into a graph here. So we're looking at timely inverted RBD specific antibody and T cell responses during acute infection. So we're looking at our acute patients. We're looking here at the weeks after symptom onset. On the left here, we're looking at the neutralizing antibody or the IgG titer and log. And then here we're looking at spot forming units of the interferon gamma ELISpot data. And then so here, by the different colors, we're looking at the endpoints of the different assays, with yellow as IgG, the green as our NAbs, red as the RBD ELISpot, and blue is the NP ELISpot. And they also show it down here. And what's important, the important takeaways here is that for your mild patients, a decent amount of them have IgG against receptor binding domain. They have neutralizing antibody titers, and they have a high RBD T cell response. So looking here at our mild patients, within week 1 and 2, we have at least some of our ELISpots coming up, but it takes until week 3 to get any kind of ELISpot data for our severe patients. And so what the implication here is that we have this delayed T cell response, which is responsible or correlated with pathogenesis. And this is despite the presence of neutralizing antibodies and IgG against the receptor binding domain. Continuing figure 5, still looking at acute patients. So we wanted to then look at the relationship between like viral load and these markers of protection. So viral loads at week 2 here are still super high. And this is despite the presence of your neutralizing antibodies. Here for the IgG and the neutralizing antibodies, where these dashed lines is just the lower limit of detection. So this is showing that yes, we are getting neutralizing antibodies, yes, we're getting IgG, but we aren't getting our T cell response for our severe patients, and that there is even a significant increase in the IgG for severe compared to mild. And there are other publications that show the same thing, that having higher levels of IgG doesn't correlate with disease protection. In fact, it's inversely correlated. And then here at D, we're looking at the viral loads, so positive or negative. And then so here we can see that there's no difference in the neutralizing antibodies, and that the RBD ELISpots is still super low. But for the mild patients who have reduced viral loads, we have an increase in the NP ELISpots. And so the conclusions here is that the NP specific T cells are needed for reducing disease severity, or at least correlated with disease severity, and the viral loads correlate with severity and the NP ELISpots. And this is really important because neutralizing antibodies did not correlate with reduced viral loads or disease protection. And again, these are with the acutely infected patients. And obviously this has huge implications in the context of vaccine development if we use neutralizing antibodies as our endpoint marker for protection, when in fact, we note here, at least the IgG, we have increased IgG in our severe patients. So that could be super important. Any comment?
They actually looked at IC50. That is, wait, is that something to do with viral growth, like, as a metric for that?
Viral growth?
Like, are they infecting cells with viruses and looking at virus titers?
Yeah, for the neutralizing antibody study.
Even though these antibodies are reducing viral titers, they're still not making the patients better. That is also strange, right?
Yes, and I'll just go ahead and give a quick plug. That is the entirety of my thesis project. So respiratory syncytial virus is notorious for having neutralizing antibodies, but not, and even a reduction in virus load, but not correlating with protection. And so that's the huge crux here in vaccine development, is that if we're using neutralizing antibodies or IgG as our endpoints, we're either going to miss antibodies that are important for disease protection but not found in the standard neutralizing antibody assays, or we're putting too much focus into these neutralizing antibodies that are not going to correlate with protection and in fact enhance disease. We see this in like dengue and Zika virus, right? So that's going to make things like antibody enhanced disease, AED, a concern for the safety and immunogenicity of vaccines for COVID.
My whole
So-
life has been a lie.
Yes, it's been a
Yes.
lot. That's why I chose this paper.
I have a comment regarding this observation. So why then the plasma treatment works with COVID patients? So basically, you're giving antibodies to a patient with COVID, and then somehow they recover. So you're just giving an extra load of antibodies, or why plasma works then?
So the antibodies that they're getting from convalescent patients may be protective. It doesn't necessarily mean that they're totally not protective. Moreover, I think if we look at the convalescent or the plasma therapy, they're kind of stepping back a little bit as far as the amount of success that they've had. I think there was a couple of reviews that came out recently that showed that people are not eager to give out plasma because of the potential of this antibody enhanced disease. Now they still are doing that perhaps as a last ditch effort, but it's not something that we would commonly want. I think that if there was a potential for convalescent plasma to be the therapeutic or the vaccine or whatever that is going to end coronavirus, that we would all be in line, right, to donate blood and to get plasma therapy. But that's simply not the case. But it is super interesting to think that perhaps there is something going on here that's even more complex than this, that if we are getting success with the antibody treatment, why that would be. I'm not sure if they're, I mean, are they doing anything with the plasma before they give it to the donor? Or are they just taking whole plasma and just giving it to them? Because I know for some IG therapies, it goes through a process where they'll isolate the specific IGs that they're looking for. So it may be that, I'm not sure.
And I have another question. I was just wondering, because here in my country, they just advertise that you can examine your antibodies for this virus, and how well do those antibodies say that you have memory or immunity for the virus? I was just wondering because some people were asking, and I was
That's a good question. So what an antibody test is going to show you is that you've been exposed to virus, that you've produced an antibody response, but the limitations is that it's not going to tell you the functionality of the antibodies, whether or not it's protective against disease. Moreover, those tests are all ELISA based, and there's not, you're not going to get a viral neutralization assay. So it's possible even that if you have high IgG, that the function of the antibodies are not there. So, I mean, here we're looking at neutralization not being one, but there are other functions of antibodies that can reduce viral loads and that can reduce disease, that have different functions outside of your standard neutralizing antibody. So the short answer is an antibody test will tell you if you were infected and if you mounted an antibody response, but its limitations is that it's not going to be able to tell you for at least coronavirus if you're protected against disease. Now, if we look at something like hepatitis, where we have Hep B, where we have pretty well understood antibody correlative of protections, things like influenza too, where there's a little bit more correlative of protection, where you're able to look at if you have a certain titer of antibodies, then you're considered protected. And that's because that's been relatively well established. But for something like COVID and either other viruses that aren't brand new, we can't make a correlation between protection and antibody levels.
Thanks. I was just wondering. Thanks.
It's also possible the real bottleneck is the T cells. Maybe you do need a little bit of neutralizing antibodies, which is there, but T cells, since they are not working as well in these patients, maybe that's what's the bottleneck for this immune response, not working so well.
Yeah, and there are a decent amount of companies now that are focused on T cell vaccines. So the review that I sent out included an overview on that technology, and I think that's going to be more and more important as the toolbox for T cells increases, and as we start to get to a wall where, regardless of how many antibodies we can induce, we're not getting protection from disease. The other thing is within these IgGs, there are probably a lot. Obviously, this is just polyclonal, so there could be, antibodies that are super protective that are just diluted out by the large amount of either aberrant or useless IgGs in there. So maybe being able to isolate a monoclonal that is particularly protective may be the way forward as well, or being able to engineer a vaccine that induces these specific antibodies as opposed to a polyclonal antibody response.
Yeah, that seems like a good thing because you have more antibodies here. You're right, could be dilution.
Mm-hmm. Diluted of the antibodies that actually do anything. And I mean, as we talked about briefly with antibody enhanced disease, inducing antibodies for a multitude of reasons can result in enhanced disease. So if you can find antibodies that are responsible for the increase of disease, you can try to filter out those antibodies induced in a vaccine platform potentially. So moving on to figure 6, we're looking at, again, the same kind of layout, except we're looking now at our convalescent patients. And we note that our convalescent patients developed our IgG and our NAbs as expected, and fewer developed T cell responses. So if we're looking at our intracellular cytokine staining in our CD8s and our CD4 cells, we have higher receptor binding domain and NP T cell response in CD4 compared to CD8. And this is interesting, obviously, because your CD8s are your cytotoxic T cells. And then, so A is showing the, did a bad job there. A, we're looking at the same data that we had seen before, where weeks after onset. And they highlighted these two patients here. And we'll, I forget why did they highlight the, oh, they were like recently enrolled or something. They highlighted why they looked at patient 39 and 40, but I forgot to write that down. Anyway, so looking at B, where we have we're looking at the interferon stimulation of our CD8s and our CD4s here, we see that we do have a reduced interferon of our CD8s compared to our CD4s. And we see that again here in this figure, taking our dot plots, putting them on this graph. And then if we look at our interferon positive CD4 positive cells for the RBDs and our NPs, again, we're looking at the subclasses that you did a great job talking about the gradient of effector memory. And for we could see the different percentages here. Importantly, most of the T cells were the EM phenotype. So by percentage. So that was interesting. And so the summary here is that we have this timely inverted response where we have induction of antibodies, but a delay in T cells.
But this is in convalescent, so this is what should happen. Like this,
exactly.
If something like this happens, people, or yeah, okay. We don't know what, like, does this mean this is the reason for their convalescence, or is it just something you see after convalescence and has nothing to do with why they got better?
Exactly. But I think what it does highlight at least is that we are getting this IgG and neutralizing response in all of our patients, but we're just not getting a good T cell response. So even in convalescent patients, so even though they survived, so then we have to ask, what's the role of protection against mortality in T cells? So I thought that was interesting. So overall conclusions of this paper. So we have our impaired DC function. We're getting a delay in our T cell response, and that's going to lead us to a more severe disease. Important takeaways here is that infection reduces your PDCs, your type 1 interferon response. You have a reduction in our NK and our T cell proliferation and function, and that T cells and not NAbs are better indicators for reduced disease and correlate with reduced viral loads. and that they identified a potential biomarker for severe disease using the ratio of CDCs to PDCs. And so with that, go ahead and see if anyone has any questions or anything that they want to show or mention.
That was a good article. A lot of observations. But they still did not show, like, did they put DCs from the patients with T cells and see if those T cells were, like, I know they showed that these DCs were not maturing, but do we know if these DCs were actually not activating T cells as much? Like they did have MLRs, right?
Mm-hmm.
I guess I forgot about that. But this is a good strategy for a virus to stop the DCs. This is an early point in activation. Then you don't have any T cells being activated.
I'll be interested once we get a little bit more information on COVID and see what the function of the virus that is responsible for the increase in MDCSs and the decrease in the PDCs. So is it something in the spike protein? Is it something in one of these open reading frames that we have no idea what they're doing to modulate immunity? So we have this mystery immunomodulation occurring. And so from a virologist standpoint, it's going to be really interesting to see what, if it's a single thing or if it's multiple things that's going on that's causing this shift in immune response.
Mm-hmm.
Yeah. Scientists don't get the right credit for the kind of complicated work they do.
No, definitely not. All they have is a bunch of gray hairs.
Yeah. And then some guy on internet shouting, I don't believe it.
Yeah, exactly. I don't know why we're getting our PhDs. We could just be selling essential oils.
Yeah. That was a nice paper. Any other questions regarding today's paper?
So I don't know who mentioned it before, but I would like to see, like in the last figure, what is going on with the myeloid-derived suppressor cells. So even like in a convalescent patient with how is the immune system like after that time, and if you have a really immunosuppressed environment, maybe this patient given another infection could also be immunosuppressed and prone to other type of infections, which it seemed really interesting. It's the first time I see this type of information.
Mm-hmm.
Yeah, it could be maybe for a future paper they could set up MDSCs from acute patients and the T cells from the convalescent patients and see then what's the T cell response. Is it something MDSC dependent or is it just something T dependent?
Yeah, it's going to be really interesting. It's also, when you think about what's causing the death here, or the mortality in COVID, we're looking at different things. So we're looking at cytokine storm, and then we're also looking at lung exhaustion, so pneumonia or something that doesn't go away. And then I wonder, like you were mentioning, what the long-term effects are, right? Because we looked at the convalescent patients, and there really wasn't a whole lot of difference in the T cells. So are we, is this going to prime our T cells for really nasty infections further on? There's precedent there, like measles infection can ruin your B cells.
Oh yeah.
So
when I read about that, I thought this is something out of a sci-fi movie.
Right?
No way.
No, it's crazy. And everyone doesn't want to vaccinate against measles because measles might not be that big of a deal. But like when you are setting yourself up to have a really bad immune system for your entire life based on an infection you get when you're a year old, like that sucks.
Yeah.
So be interesting to see. And then the context of reinfection and protection. Can we make any correlation between a T cell response and protection against future infections? Maybe it is these high antibodies that we have that might be able to prevent infection from occurring in the first place, or maybe it makes it easier to get infected. Your disease outcomes worse.
Yeah. Who's making these cytokines if everything is suppressed?
That's another good question. And I saw a paper recently, and I should have saved it, but it was talking about, instead of it being a cytokine storm, that there were some other effects going on that's causing this, and that cytokine storm was kind of like the easy the easy culprit to blame, and that there's this other storm that's occurring. Mitokine, is it the mitokine storm? That's not a thing, but it's something like that.
That's just immunologists trying to make fancy names for things as they go along.
Yeah. No.
There's a question from in the chat. Carlos Alfonso, he asks, could this be a finding bias? They look for T helper 2 response, but it may be that all T helper subsets are increased, or it could be just a correlation. Did they look for T helper 2 cells in this? Like, did they stain for any T helper 2 markers?
I think they just did CD8 versus CD4s.
Okay, yeah, maybe it could be a specific type, a polarized subset of T cell that is doing, and while everything's going down, that one subset is going up. That is possible.
Yeah, what they probably should have done instead of doing a single plex ELI spot is do a dual color ELI spot where you look at interferon and IL-4.
Yeah, they did a lot of work for this. If I would be a reviewer too if I told them not this is not publishable.
Oh my gosh, right? Like, I don't know what the draft looked like, but this paper was so much work. I couldn't imagine putting this into Cell and someone giving, no, I want you to look at the T
Yeah.
response too. It's like, okay, whatever, dude.
Your life sucks.
Yeah,
Just send it somewhere else.
Yeah, exactly. Like, I have all of these data. You're getting hung up on the TH2 not being there, but I think that would be super important, and that could have been super easy to do and give them, I mean, a whole new idea of the role between TH1 and TH2s in this.
Yeah, they're probably going to pursue that. Hey, thanks, Carlos, for the question. Who wants to go next for our journal club? It's going to be on 26th September. Anybody wants to volunteer? We can also discuss in the Slack. But yeah, probably going to be 26th September. That's 2 weeks from now.
I could try. I have one article about some changes in some like glycosylation receptors, and they investigated it in mice and in some form of Sjogren syndrome model, and they found that it was somehow age-related. So
you said
yeah,
Sjogren's, that's autoimmunity. I'm already in for it.
Yeah.
I'm in for it. I have a very explicit bias for autoimmunity. Yeah, all right, let's do it. 16th September, it's no 26th September, Maria's turn.
All right, well, thank you everybody for tuning in today. Thank you for
awesome presentation, Harrison.
Good presentation, Harrison.
Thank you guys. Thank you.
And everybody watching this, thank you guys for joining. I'll end the live stream now.
All right.